So, quick show of hands. Who has heard the word GMO? I'm sure many of you have. GMO stands for genetically modified organism. And chances are you probably have some GMOs inside of you right now. Why? Because most of the GMOs we make involve plants that we grow and harvest for food, ranging from things like tomatoes to soybeans, strawberries, and most of all, corn. So what are GMOs? So what is a genetically modified organism? We will define a genetically modified organism, or GMO, as any organism that contains extra genes from another species. So in other words, any organism that contains bits and pieces of DNA, genes, that came from a different species. So an example of this, scientists have actually created genetically modified strawberries sold in grocery stores that contain a gene from this guy right here, an Arctic flounder. Arctic flounder live in freezing cold waters. So because of this, they have a gene in their DNA that codes for a protein that prevents their blood from freezing. So this is like an antifreeze gene found in the Arctic flounder. These scientists took that antifreeze gene and put it in the strawberry. The result are strawberries that are freeze tolerant, unaffected by frost. So if you have freak storms like a snowstorm in April when you're hoping to be growing and harvesting planting your strawberries, or a freak early snowstorm in October when you would hope to be harvesting your strawberries, they might be able to survive that and you get a longer growing season. So scientists can make this happen in the following way. So imagine we have two cells. Cell A from one particular organism and cell B from an entirely different organism, right? Here's cell A's chromosomes. It's got 3 chromosomes you can see right there. Cell B has 1 chromosome, but that chromosome has a gene that makes it glow neon green. So when that gene is turned on, it gets transcribed into mRNA and translated into a protein. And that protein makes cell B a fluorescent neon green color. Now, what scientists do in genetic modification is that they take this gene from cell B and they put it into cell A, like that. Now, with this gene now in cell A, if it gets turned on, it will get transcribed and translated. That's again transcribed into mRNA, then translated into a protein. And that protein will make cell A glow bright green as well. Neon green cells. So you took the neon green gene from cell B, put it into cell A, and made cell A glow neon green as well. So one thing that I would hope you would take away from any biology class is the fact that bacteria are cool. Bacteria are actually really amazing. And they're some of the coolest organisms on Earth, except when you find the wrong kind in your Chipotle burrito and you spend the next 24 hours on the toilet. Then they are probably not very cool to you, but still cool from an objective scientific perspective. Bacteria, one of the many reasons they are so cool, is that they have the ability to do what we just saw on their own. So let's say we've got a bacteria and it's swimming along through its little microscopic drop of pond water, and it encounters a piece of DNA floating in that pond water with a gene right here that codes for a neon yellow color. This bacterium has the ability to encounter this gene, swim up to it, take this piece of DNA, and actually suck it up inside itself. So now the bacterium has a piece of DNA containing a gene that codes for neon yellow color. If this gene is turned on in the bacterium, it will be transcribed into mRNA and translated into the protein that makes neon green color. And as a result, this bacterium will glow, neon, I said yellow, but I drew green. We get the picture, right? The bacteria picks up the DNA from its surroundings, and it, in turn, glows. So this process is called transformation. Transformation is when bacteria eat up DNA from their surroundings like we just saw. They encounter a piece of DNA in their surroundings and they suck it in and make it their own. Now this is very useful because often what the DNA that bacteria are encountering aren't coding for things like neon yellow color. When bacteria die, they leave their DNA behind, and sometimes that DNA can stay useful. So bacteria, when they're all swimming around, might encounter DNA from another bacteria that figured out a way to break down a new food source or break down a certain toxin so that it could resist poison. This DNA, even though that bacteria had died and left it behind, would be extremely useful to another bacteria. So bacteria have evolved this way of picking up DNA from their surroundings and then putting it to their own use. So that's all really cool. So that might bring up the question, if bacteria can do this, and we can do this with things like strawberries and tomatoes and corn, can we do this with people or other animals? Now, there are proteins out there. There are genes for proteins that actually do result in a neon green color. Could we put this gene in a person and make that person neon green? Well, there would be a lot of ethical objections to this, so that wouldn't really be a question with people. But what about other animals? Well, the answer is yes, but you have to do it in a certain way. So here's a pig. And let's say that we want to make this pig glow in the dark by giving it a gene from a firefly that allows it to produce this flash of light on those warm summer nights. That ability is coded by a gene in the firefly that produces a protein which can cause the chemical reaction which makes it flash light. So couldn't we take that gene from the firefly and put it into the pig? Well, theoretically, yes. We could take that gene and put it into one cell in the pig. But the pig is made up of anywhere from 100 to 150 trillion cells. So in order to make the pig actually glow, we would have to very painstakingly take DNA from a single cell in the firefly and put it into every one of the 100 trillion cells in this pig. That would be impossible. What we can do instead is take that gene that codes for the flash of light and put it in, not an adult pig, but a pig at its very earliest stage, a recently fertilized egg, also known as a zygote. If we take the gene from one of the cells in the firefly, isolate the gene for making the light flash, and place it in a pig embryo, that embryo will then start to divide and make copies of that gene, and more copies, and more copies, until eventually every single cell in the growing pig embryo contains the gene for that flash of light. So, what will you get? A lovely, mature firefly pig. And that is how you make a GMO, or a genetically modified organism. Now, keep this stuff in mind because we will be creating our own genetically modified bacteria in the lab in the coming days. And we will use that process of transformation in order to do so.